Hey, so welcome back, and today we would be talking about monohybrid cross. So what exactly is a monohybrid cross and what do we do in it? That is something that we would be talking about today. So let's just start the work. So when you talk about a monohybrid cross, mono means one. And hybrid means it is happening between two different types of the similar plant. So mono means one, hybrid means between two. So these two plants that we are going to talk about, they differ from each other in one particular character. That is what is our assumption. In cross, because it is happening between the two, so that's why a cross. So now let's just start with what is a monohybrid cross. So now we shall start with the monohybrid cross. So first thing we have is our parent generation and their genotype. So parent generation, the first one plant that we are talking about is this, a tall plant, and the other one is dwarf. Now we need to keep one thing in our mind, which is both the parents, they should be in homozygous condition for the trait that we wish to compare. And if we have to write their gametes, then as I said earlier, that gene pairs, they split apart and they end up getting into different gamete cells. So this will end up in a separate gamete, this is going to end up in a separate gamete. Similarly, this one as a separate one and this one as a separate one. So now we need to work out on their possible combinations. So what is going to happen if someone meets with someone, or let's just see what happens when who meets whom, we can say. So for this, I have a simple thing here. We can make a box. Let's just make this box. Okay, so I'll split it into 4 parts right now because we have 4 gametes. So I'll write the genes of one parent to this side and the genes of other one to this side. Now once I start filling, I'll pick up this capital T and I'll put it here, and then the small t, I'll put it here. I'm putting this one here, why? Because I'll tell you why, just wait. And then this and this, this and this, this and this. So now I know that this has happened. So if I need to write the genotype of my F1 generation, I can say the set of genes that I got are these, capital T and small t, capital T and small t, 4 times. Now depending upon this genotype, if I write the phenotype, which is the physical appearance, so I know that this trait is going to express itself. Tall, 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 tall. So in the F1 generation, a new term here, F1 means the kids of these parents. F1 generation, we see all the plants are tall, which means dwarfness or short height. It completely disappeared in the F1 generation. Now that's something which is not a magic. It is because tallness is dominant. And the trait which disappears in F1 generation is known as recessive trait. So small t and small t is, I'm circling it here, it is recessive trait. So the trait which expresses itself in F1 is dominant, and the trait which vanishes away or which does not show up in F1 is recessive trait. So I hope it's clear as of now. So now what we do is we talk about the selfing of F1, which means the genotype of my F1 plant was this and this. Now here you need to understand one thing. When I say the genotype of my plant is this, it means the complete plant, all the cells of the plant will have this type of genes. So be it anther or be it ovules, they're all going to have this type of genes in them. So similarly, we are going to split the genes apart now. So half of the anthers are like this, another half are like this. Ovules are either like this or they are like this. So this is how we make the gametes. Now since we know the gametes, so now we can take it further. Sorry, genotype, yeah. Okay, so let's just start with the F1 cross now. So in the F1 cross, the cross of F1's selfing actually, we can see that the genes this time are a bit different. So we have a capital T and small t, capital T and small t. And please be assured that the trait which is dominant is denoted by capital letter, and the one which is recessive is denoted by small or lowercase letter. I'm sorry, I forgot to tell you that. So now make sure you do not confuse that similar genes should be together, so it should not be like this. That is not correct. So here we have capital T and capital T, and here we have capital T with small t, capital T with small t, and small t with small t. So now the genotype, the set of genes that we get here is this. We get this. We get another one like this. And then we get the other one like this. So phenotype, capital T, we know it's dominant, tall. This one's again dominant, so it is going to be tall, tall, dwarf. So this clearly shows that a recessive gene was present in F1 generation. However, it did not show up at that time. So if we need to work out on their phenotypic and genotypic ratios, how we are going to talk about that. So for our reference, what we can do is I'll quickly write down the genes that we got. We got this, we got this, we got this, and we got this. So we got 1, 2, 3 tall, and the other one as short. So we can say phenotypic ratio is this, which is 3 tall is to 1 dwarf. How about if we talk about genetic ratio? So we call this condition as pure tall, this one is a hybrid tall, and this one is a pure dwarf. So we have one which is TT, two which are like this, and the one which is like this. So 1 is to 2 is to 1. 1 pure tall is to 2 hybrid tall is to 1 pure short. So this is how we say is the genotypic ratio is. It is 1 is to 2. So this thing tells us about what is a monohybrid cross, and this thing gave us the first law of inheritance, that is the law of segregation. So it says that the alleles of a character, though they remain together, however, they separate in subsequent generations. So these, or maybe these, though they remain together, but then they separate in the subsequent generations, or probably at the time of gamete formation. Clear? So we'll do it till here only, and keep watching this area because soon you're going to get the next video that talks about dihybrid cross. Okay, so see you guys later.